I would also like to acknowledge that today I'm on the traditional land of the Bindal people and that I pay my respects to elders past and present. I also wanted to say thanks to the ACRS for the award. I was really honoured to receive it. And ACRS, I would say, is one of my favourite societies. I remember those student conferences and how great it was to go as a PhD student and how you could talk to anyone. It was really easygoing. And then I also enjoyed my time on council as well. And so I know everybody who's joining today knows what a great society it is, but I hope to see you all next year for an in-person conference. So yeah, I'm going to talk today about my research that I started during my PhD on thermal plasticity of tropical marine fish, and then maybe finish with just a little bit of what might be coming up in the future, which is research that's been led by my students. So I'm sure everyone that's here knows that we have a concerning problem at the moment with the human population continuing to burn fossil fuels, releasing over 36 billion tonnes of CO2 in 2019. And the concern for this increasing CO2 is that it's enhancing the greenhouse effect, which means that we're trapping more warming within the Earth's atmosphere, which is causing rapid warming beyond the normal conditions. And why we're so concerned is that organisms have evolved to the climatic conditions that they've seen historically. And just one of a graph on the right, which is by NOAA, shows that, you know, from 1880 up until approximately 1980, we're seeing conditions that are quite different to this rapid increase that we're now experiencing in terms of global land and ocean temperatures. And this is quite a big shift for the organisms that live on the globe. So we're particularly concerned about ectotherms, and that's because species that are ectothermic don't have the capacity for internal temperature regulation, which means that their body temperature and the environment are quite closely linked. So for the tropical oceans, we're expecting that we might see warming up to 2 or 3 degrees Celsius by the end of the century. And we know that tropical species have evolved in quite thermally stable environments, so there's not a lot of seasonal variation that they experience. And we also know that tropical ectotherms are living quite close to their thermal maximum. And everybody here today knows that we're particularly concerned about coral reefs and the corals that make up the reefs, because they're likely to be one of the ecosystems that's most severely affected by this ocean warming. And so what we know about tropical reef fish at present, so one of the ways that we often view how an animal or a plant responds to temperature change is through what's called a thermal performance curve. So we've got temperature here on the x-axis and a measure of fitness or performance on the y-axis. What we find for tropical reef fish is that as water temperature increases, it goes up to an optimum. And then once we pass this thermal optimum, we see a decline in performance. And for tropical fish, the research we've done so far suggests that their thermal optimum is pretty close to the summer temperatures that they experience. And when we take fish from the reef and bring them back to the lab and put them under an increase in water temperature, we generally find this decline in performance. So they're definitely sitting at the optimum. And as we apply further warming, we see this right-hand side of the slope where fitness and performance declines. And so things like growth reduces, reproduction, swimming ability, and even some things like sex determination will be affected by these increases in temperature. But this is just the current day response. This is just the short-term response of a given population at the moment in terms of how they respond to the temperature increases that might be occurring by the end of the century. So there's a couple of ways that organisms could respond to changes in their environment, and one of which is through adaptation. And adaptation occurs through natural selection and selection of heritable phenotypes. We know that selection can occur on either pre-existing genetic variation when the environment changes, or it could occur on top of mutations. But what we do know is that selection requires time, and thus this may be a problem in terms of the rate of rapid climate change. And often when we think of adaptation, we think of it at the species level. So this is just an example of an ancestral finch. And through time, natural selection has caused this one species to split into three, depending upon the prey items that they've consumed and shifts in their beak morphology. But really, in terms of climate change, we're interested in it at the population level, where as the environment shifts, perhaps there's selection against, in this case, the bluefish, and the resulting population is then better fit and are better performing in those future conditions if this variation can be heritable through time. But as I said before, because adaptation requires natural selection, it requires multiple generations and it requires time. So there's generally been the concern that for most species on the planet, adaptation will take too long to be the response to climate change. So the other way that organisms can respond is through phenotypic plasticity. And this is the ability of an organism to change its phenotype in response to changes in the environment. Two examples on the right here are, the first is the water flea Daphnia, where when there's the experience of predators, they make these little spiky helmets. But if there's no predators in the environment or no, response to the predators, then they don't form the helmet. In the case of butterflies, we see plasticity across seasons. So if there's a wet season, the left phenotype with brown and smaller wings forms, whereas if there's a dry season, this different wing morphology and color forms. So this is all non-genetic. So in this case, these are possibly even the same genotype responding to that environmental stimuli. But we do know that genetics plays a role. So whilst it's non-genetic, the underlying genetic architecture or the genotype can still have an influence on how much plasticity occurs. There are limits to plastic responses, but definitely phenotypic plasticity has come up a lot in conversations because the rate of climate change favors this particular response. So really, where my PhD and my research started on this topic was, well, can warming induce plasticity in coral reef fish? So there's three main types of plasticity that we often talk about. The first is reversible, which is a response within a generation, generally to daily or seasonal environmental fluctuations. The other is developmental, which is again a response within a generation, but it generally occurs due to experience during early life. And the belief is that these developmental responses are quite permanent. So if you experience a predator early in life, you get that helmet, but you don't necessarily lose it if there's no predators there in the future. And transgenerational plasticity is the influence of parents or previous generations to affect the offspring phenotype. And so what we know for coral reef fish is that reversible plasticity seems to be quite limited. And this could be because generally tropical species don't experience that much seasonal or daily fluctuations when we consider it across or in comparison to temperate species. But to me, that's okay, because we know that climate change is going to shift the environment both within and across generations. So there's still the potential for developmental and transgenerational plasticity to play a role. So I started off my research with the spiny-chromis damselfish, Acanthochromos polyacanthus, and the population that I've mostly worked with is from the Central Great Barrier Reef, from the Palm Island region. Collected breeding pairs and brought them back to the lab at the aquarium complex here at James Cook University, and simulated the current day temperature +0, or often refer to it as, and what might be expected by the middle or end of the century, depending upon what we do with emissions, of +1.5 and +3 degrees Celsius. And so the temperatures that these fish experience was a seasonally cycling temperature. So we didn't keep them at summer temperatures for the entire year. We tried to make it a little bit realistic. And today I'm going to show you sort of two main groups of traits that I've looked at over the generations. The first is aerobic physiology. So this has become quite a popular metric because you're able to measure it on live animals and measure it multiple times. So you could take a single individual and look at how that individual responds to temperature in terms of their metabolic rate. And so what generally happens is that we've got temperature on the x-axis again. is that as temperature increases for a fish, the standard or resting or routine metabolic rate, it gets called a range of things, tends to increase as the water temperature increases. Now, the maximal metabolic rate, which is the maximum capacity to deliver oxygen around the body and to make energy through those aerobic processes, also increases as temperature increases to a certain point, and then the cardiovascular system doesn't seem to be able to keep up. And so what we see when we think about the difference between the maximum and the standard is that this gap starts to narrow as water temperature increases. And it's this difference between maximum and standard that often gets called aerobic scope or aerobic capacity. And to do this, we measure oxygen consumption as a proxy. So this results in aerobic metabolism or aerobic scope, showing that sort of nice thermal performance curve like I discussed before. The other set of traits that I focused on looking at is reproductive capacity. And this is a really interesting thing to understand because it obviously directly relates to population sustainability and fitness. We also know for most tropical coral reef fish, they like to spawn in spring to summer. So it potentially puts them at really high risk with the ocean warming. And we know that reproduction in most species is optimized to a really narrow temperature window. So they don't tend to reproduce at much cooler than that, and they don't tend to reproduce any warmer. And so I measure things like the spawning season length, how many pairs reproduce, what amount of eggs and what size eggs, and then the quality of offspring. And when we come back to this sort of testing the acute effects of temperature, so bringing fish from the wild into the lab and simulating ocean warming, we see that there's definitely negative effects of almost all these traits with ocean warming. So reproduction is highly sensitive. So to start off, what we know about developmental plasticity first. So the experiment started with a wild population of adult breeding pairs that then produced clutches within the lab, and I then split them between present day control, plus 1.5 and plus 3 degrees Celsius. So in this generation here, we have sibling fish that have just experienced different water temperatures throughout development. And I'm gonna, instead of overwhelming everyone with a whole heap of graphs, is just present the summary of what we found in these tables. So just a recap of what the acute responses are. So as water temperature increases, we see that standard metabolic rate increases, we see that aerobic scope decreases, and we see that all the traits we like to measure related to reproduction also tend to reduce as well. Well, with developmental plasticity, we see that most of the physiological metabolic traits seem to also remain the same. There's some partial improvement in terms of standard metabolic rate in the group that experienced plus 3 degrees Celsius throughout development, but it doesn't bring it back to sort of what you would expect with full restoration to control levels. In terms of reproduction, I now see that there's more capacity for developmental plasticity in terms of reproductive traits within this plus 1.5 group. So they're able to produce clutches of a similar size, provision their offspring with a similar amount of yolk. And there's also a similar amount of breeding pairs reproducing in the experiment. And in the case of plus 3, we only really see a shift in terms of the amount of yolk provisioning. So it seems that there's some capacity for developmental plasticity, but it might be quite limited. I also just wanted to touch on when I talked about developmental plasticity, that it's probably not going to be a simple matter of studying one population or one species. So we've also looked at the differences in terms of developmental plasticity of aerobic physiology by comparing two populations of the Spinochromis, one, the same one I've already spoken about in the middle central Great Barrier Reef, and then another population from Heron Island. And we find really different patterns between these two populations. So in the short term, we see that there's really similar responses. So this is resting metabolic rate or routine metabolic rate or standard metabolic rate. As water temperature increases, they tend to increase at a similar rate regardless of the population, whether it's from Heron in the black or Palm Island in the grey. But their capacity to deliver maximum oxygen seems to be quite different. So the Heron population is able to deliver a much greater rate of maximal oxygen. And this results in still in a decline with net aerobic scope overall. So When in the short term, we don't really see any differences between the two populations. However, when we look at their capacity for developmental plasticity, we see that this black line here has come down really, really low, which means they've basically reduced the cost of daily metabolic function at those warmer temperatures. And that's meant that they're able to basically restore their aerobic capacity. So in this case, the Heron Island population had a very high capacity for developmental plasticity in comparison to the previous Palm Island population. And we do know that the southern part of the Great Barrier Reef for this species is quite genetically distinct. So this could be due to differences in the underlying genetic architecture. I've also looked a little bit across species and found that there's real differences for developmental plasticity depending upon which species you look at. So across all the wrasses we've studied, it seems that developing at these warmer conditions actually is worse. So you see even poorer performance than if you just tested them at these warmer conditions in the short term. We see that Pomocentris mollikensis, plasticity was quite similar to Acanthochromos. It was possible at moderate levels of warming, but not at higher. And in the case of the spine-cheek anemone fish, we see that actually the performance improved with all warming. So they seem to like it a little bit hotter than all the other species we looked at. And so this does show that there is going to be diversity across coral reef fish. It's just a bit of a challenge to work with most of these in captivity for long, long periods. So what about transgenerational plasticity? Well, we know that there'll be periods of gradual warming through time. And so now we're trying to add in simulating what it'll be like if the warming is experienced slowly across generations. So it's the same design as before. I have all those fish that I already told you about with developmental plasticity in the first generation. They then reproduce and we split the clutches between different conditions. So now here, this is the same developmental group. So, but in the F2 generation. So I have the acute present day, those that just experience warming from hatching. I have my transgenerational groups that experience 2 generations at plus 1.5, highlighted here in the yellowy orange, and 2 generations at plus 3, highlighted in the red. And then also we now have this step treatment where the first generation experienced plus 1.5 and the second generation experienced plus 3. And the goal with this particular group is to try and understand if we get the same response when they experience warming gradually across populations or when they just experience plus 3 degrees Celsius within a generation. So also to look at transgenerational effects to reproduction, it gets a little bit more complicated. So now you'll see that there's this box around the fish, and that's basically to indicate the reproductive environment. Because we know that reproduction is so temperature sensitive, we need to understand the effect of developing in these warmer conditions separate from reproducing in these warmer conditions. So here you'll see that the blue fish, these are all the control present day fish, and they just reproduced now at plus 1.5 and plus 3 highlighted. In terms of the plus 1.5 treatment, we've got those fish that have developed for 2 generations at plus 1.5 and then reproducing at plus 1.5. We've got the plus 3 group, 2 generations at plus 3 and reproducing at plus 3. And then we've got these 2 different groups here. One is the step. So we're seeing how the step group reproduces at plus 3. And then this group here is asking, well, if you spend 2 generations at plus 1.5, does it provide you in a benefit if then you have to reproduce at this warmer condition of plus 3? And so here's the same table that I showed you before, now just blown up. So just to recap, we see some partial improvement in terms of standard metabolic rate with developmental plasticity at plus 3 and a little bit of restoration for reproduction. We see multiple traits restored at plus 1.5 in terms of reproductive plasticity. And just starting off with these 2 groups that are the straight transgenerational line. So there's no change in temperature with the reproductive environment. And now we see that actually 2 generations at plus 1.5 allows restoration of these physiological traits. So if both you develop as well as your parents in these warmer conditions, there's an improved response. Clutch size, offspring yolk area, and proportion of breeding remains restored as we saw in developmental. Now, in terms of the group that experienced +3 degrees Celsius for 2 generations, we now see that actually their aerobic capacity is completely restored. So their physiological traits have restored within 2 generations. But in these fish, we saw absolutely no reproduction. So there does seem to be a mismatch in terms of the capacity for plasticity and physiological traits, or aerobic physiological traits, and the capacity for plasticity in terms of reproductive traits, which is really interesting. So these other 2 groups here, in terms of fish that developed at +1.5 and then reproduce in these warmer conditions, we actually see that developing there doesn't necessarily provide you any benefits if it continues to warm, if you're only experiencing those warmer conditions during your adult life. But if you're experiencing these warmer conditions from development, so in the step treatment, that the first generation is at +1.5 and the second is at +3, we see improvement and restoration in the aerobic capacity, and we actually see a lot better reproductive capacity. So it seems that perhaps for some of these reproductive traits, experiencing warming during early life is definitely quite important. So the other angle that I wanted to talk about today was thinking about, okay, that's great. If it's just continually warming into the future, those fish will be better capable of responding with those warmer conditions. But there's obviously definitely limits depending upon how much warming they experience. Well, what about if you experience periods of warming that are then followed by periods of normal temperature? Does plasticity last? Does the effect of this generation experiencing the warming then benefit when a future generation also experiences that warming? And this experimental design has just focused in on the control in the +1.5 in this case, because the +3 just didn't reproduce, and to try and cross-factor and understand, is it critical that your first generation here or your parents and grandparents have experienced the warming to get benefits to this third generation, or would it be fine if they just experienced it during reproduction? And so again, the fish color is the temperature that the F1 and the F2 generation developed in, and the boxes are still the reproductive environment. So we have a pretty much almost perfect split within the third generation, where every group it develops that control or +1.5, but unfortunately one group's missing. And so I'm just going to tell you about what happens in this third generation when all the fish are back at control levels. So the important thing here is that this third generation has never experienced warming themselves. Any influence that they have is based on whether their parents or their grandparents experience the warming. And I'm only going to present aerobic metabolism. So starting off just slowly, I've got routine metabolic rate on the y-axis, and now we've got the parental reproductive temperature. So this is the color of the boxes here, so whether they reproduce at +0 or +1.5, and the blue line is the blue colored fish. So the control line is showing that as parental reproductive temperature increases, there's a slight increase in terms of what then the third generation's routine metabolic rate was, but it's not very big. But if your parents experience those, parents and grandparents experience those warmer conditions, there's definitely a strong effect of whether your parents reproduce in control or reproduce in +1.5 conditions. Routine metabolic rate was a lot higher if these fish experienced their reproduction at +1.5. In terms of aerobic capacity, we see that for the control line, there's not really an effect of parent reproductive temperature. So simply by having a heat wave during summer when the parents are reproducing isn't necessarily shifting this aerobic phenotype. They pretty much look the same as the siblings. But in the case of the +1.5 legacy group, we see that there's an increase in aerobic capacity overall. So if your parents or grandparents experience these warmer conditions, something has shifted that then means regardless of whether you experience a warm condition, you have an increased aerobic capacity overall. So this is what's required to sort of prove that what I've seen before is truly a transgenerational effect. So then I just wanted to quickly touch on some work that I've really been lucky to collaborate with others on. So this is very much not my expertise, nor is it anything that I've been able to lead. But I guess the thing that these experimental studies do is allow us to really go into detail because we've controlled everything else to try and understand the effects of temperature and, in this case, what the mechanisms might be at the genomic level. So this is work that's been led by a collaboration with Tim Ravasi's lab, Heather Tewu and Moises. And I'm going to just quickly talk about some of that work to show you what you can do with, these detailed experiments. So to start off, to understand the mechanisms of plasticity, we've been focusing on the liver as the main tissue. And Tim's lab has then gone through and assembled the genome for this population. And looked at differential methylation in certain cases. So we don't necessarily know as a scientific community what methylation does, but one of the ideas is that by having methylation, you're turning off a gene that is no longer used, or by removing methylation, you're then activating the access to a gene that might be required. We've also looked a lot at the transcriptome, so looking at which genes are expressed between these different treatment groups as a way of maybe understanding how they might be improving their aerobic physiology or improving their reproductive capacity. And so definitely don't want you to look at all the details, but this is work by Heather that was our first step into trying to understand what genes were differentially expressed when these fish experienced development at the warmer conditions or transgenerationally experienced the warmer conditions. She's then gone through and highlighted sort of the likely functions that these genes are involved in, categorized them. And then we've also looked at what percentage of the gene expression seems to correlate with the physiological metabolic traits that we've looked at. And there's a lot of detail here, but the really thing that I find most interesting is that this is a suite of gene names on the side here. And when you compare the developmental and the transgenerational response, we weren't seeing that a completely different set of genes was being upregulated. It was generally just that the response of the transgenerational group was much stronger. So the differential gene expression was much stronger. Moises then went on to look in detail at this step group to try and understand what differences existed if you experienced the warming slowly over generations. And here he found that actually the signature is quite different with the step on the right-hand side in both cases. Again, there's no gene names here, but this is just showing pattern of differential gene expression that is quite different between whether you experience a really rapid change in temperature versus if there's a gradual change in temperature. Then Tewu sort of investigated the methylome a little bit more. And in this case, we've had to narrow down just to this +3°C groups, just due to cost and ability to understand everything. And he's gone through and looked at differentially methylated regions and found that 51 of the genes that showed a differentially methylated region also matched what had previously been found in terms of differential gene expression, and that 7 of those genes seem to correlate to the aerobic performance that we'd investigated. So this is pretty cool. Like we don't necessarily know exactly how it happens, but it seems like we're starting to interrogate and understand more maybe which genes are really involved in these transgenerational responses. And most of the time when we think about gene responses to temperature, we think about heat shock proteins. And interestingly, throughout all these investigations, we don't see that heat shock proteins are involved in these longer term responses to temperature and how fish will likely acclimate over multiple generations and in the longer time scale. So I just wanted to summarize what I think we know so far. I think we know that plasticity has the potential to play a large role in responding to climate change and in this case, ocean warming. But we definitely see that there are limits if the warming is too extensive. So minimizing emissions do matter. And we're hearing that more and more with all the rest of the coral research and keeping it under these certain threshold temperatures. Definitely see that reproductive plasticity is more limited than physiological. And I think that does indicate that we need to make the effort to do generations for long enough, or to do experiments that allow for understanding reproductive effects. As much as I love to focus on my one model species, I have to admit that it's going to be complex within and across species. And so we do also need to make an effort to study, outside of one particular model organism. But I do think that understanding the few model species well really is important to maybe breaking down some of the mechanistic understanding that we could then apply to other species we'll never be able to study in captivity, or we just don't have enough time for everything. And I didn't present it today, but we do have some research linking whether plasticity could be adaptive, but definitely this is an area that we haven't necessarily done enough on, and should be an endeavor for future research. And then I just wanted to talk about, after this sort of first experiment, where we've been going with everything. And so this is very much now not so much my research, but the research of the students within the lab. And some of the cool things that we've done is that we've added a diurnal temperature cycle into the experiment. So when we started my PhD quite a while ago, our capacity to control and simulate was a bit more limited, whereas now with the technology advancements, we've been able to put both a seasonal and a diurnal cycle. We're finding that that's actually shifting things quite a bit from what we might have expected from my experiments. We've been looking at the roles of mothers and fathers in transgenerational plasticity, and this is work led by Rachel and Lucrezia. And a lot of this will be coming out shortly, hopefully in the next sort of year or so. So this research is finished and has just been worked up. In terms of the future directions, we're looking at how heat wave exposure impacts thermal plasticity. So much shorter exposures than I've ever used before. How does weeks or a few weeks of thermal exposure maybe influence the capacity for fish to then respond when they see that warming again within their lifetime, or even across generations? We also realized that perhaps the local adaptation, especially in this species that's already occurred across the Great Barrier Reef, could be really pivotal in terms of their capacity to respond to future warming. So we know that Heron Island population had a much greater capacity for within generational plasticity. And there's definitely a need to understand, do we have certain populations that are much better primed to be ready to go with environmental change? Or is it, a much more complex story than that? And we're also interested in understanding how transgenerational plasticity will influence how fish respond to other stresses like ocean acidification and sedimentation. And so classically, when we've done these experiments, we've always done them together in tandem, but we're now interested in understanding, if you experience warming first and then layer another stressor on top, does that influence your capacity to respond to climate change? And then finally, I definitely have to say thanks to all the funders that have made the research possible over the years, and thanks to you guys for watching.
Wow. Thanks, Jenny. I think I can speak for everyone in how good that presentation was in really going through probably the most comprehensive set of experiments from their inception from your PhD over a decade ago, and just such a well-told story the whole way through. It keeps growing and developing, and it all happens with reason and purpose, and it's amazing to see all together like that. So thank you. I'll be happy to mediate question time. So feel free to unmute if you have a question, or post it in the chat otherwise. I'm happy to start us off. One thing that I've been just thinking about through this talk and recently, with all of the coral reef restoration and kind of transplanting corals that are maybe more tolerant thermally or whatever the reason is, in all your research, trying to key in on some of these like genetic or phenotypic adaptations, do you think it's at all possible that we would also start seeding thermally tolerant fishes onto the reef in some capacity?
Yeah, I'm not sure that, I mean, fish are a bit sneakier, right? Like, if you move them, they have this mobility option. So maybe they'd also not do what you wanted them to do. But for me, I'm not convinced we're at the point where we have enough information to start switching and swapping, because I think as much as we expect that, say, the populations that live close towards the Torres Strait, if we move them down south, we pre-prepare and they'd be ready to go. But we actually haven't investigated enough to know that that's true. So, are these populations actually more thermally adapted? And then I think there comes the complexity of, well, when do you move them? Because if you were to move them early, I guess plasticity could work against you in terms of they could just get used to it being cooler and all those things you thought you'd primed for didn't happen. I'm kind of more interested in whether we could know more about the diversity that exists in the genotype phenotype relationship across populations, and is there a way to think about that in terms of how we conserve and manage. So if we know that certain populations have very high capacity to cope with environmental change, maybe because they've just naturally experienced more daily variation or more upwelling events, or because we know that there's reefs that bleach a lot more than others, so we know there's variation. Maybe that's a better way forward in terms of protecting what we know would give us the best chance of having that species in the future.
Thanks.
Maria's got her hand up.
Yeah.
Jenny, great talk. Now, I noticed that, in the genetic work you said wasn't yours, but you said that, I know Heather's work and the heat shock protein, everyone jumps on that, but there's a lot of other genes that are really important as protective products. Did you find anything else in the gene array, ubiquitin or ROS or any other responsive gene that might indicate a marker to look at?
I think I would have to divert that one to Heather in terms of my ability to remember all the genes. But I think that it's more just that you're right, everybody jumps on heat shock proteins. And I just wanted to mention that probably is much more involved in the acute response in terms of how fish will deal with things like heat waves. But I couldn't probably comment on all the genes that were there, because that's definitely more her forte, unfortunately. Sorry.
That's all right. No, it's very interesting.
You're on mute, Kenny.
So everyone, I'm on mute. Classic. Haven't done this enough times. I think Sven, you had your hand up next.
Yeah, hi, thanks. Just chiming in, that was a fantastic talk, Jenny, and thanks. I'm just jealous of that you could do all this. As you know, I did a little bit of this over short generations with urchins. I'm a bit, hang on. I'm just equally mean as Maria. I had a question about the genetics. Sorry, but you don't need to answer. I was just curious because you mentioned the methylation patterns, and that most likely methylation patterns reflect whether genes are switched on and off. I just wondered if you looked at some sort of correlation between gene expression and the methylation, because if it's switching genes on and off, there should be a reasonably clear relation between it. So
yeah, so Tewu definitely did, and I think there was it was the 51 genes that I mentioned. I think in all our conversations in the group, the collaborative group, the challenge is maybe about timeline. So often we take these samples when the fish are big enough and the tissue's big enough, but by then they've spent usually 2 years in these warmer conditions. So I guess there's a challenge that we're looking at the wrong window when the methylation and gene expression is really playing a role. So maybe it's much earlier in life in that developmental window. And if we went in on that area a bit more detailed, we might better understand, because I think most likely we're missing some of the picture, if that makes sense. We're sort of underestimating the understanding between methylation and gene expression, because the gene expression may have already occurred during development, and then it's not necessarily something that's very clearly upregulated later in life once the developmental phenotype set.
Yes, I think, yeah, thinking about it, I think you're right. There shouldn't be a clear relation because it's different time scales, isn't it? Because expression can even be different whether you sample at lunchtime or in the evening. Yeah, so maybe my question wasn't the brightest.
No, I think it highlights how interesting methylation is, but how little we know. And most of what we know is from these really tight model systems, you know, in rodents, and where they've managed to get clones and they don't have all the other chaos that we probably are seeing as well in terms of differential gene expression. But if it was a little bit cheaper, I think we'd do it more often. It's just hasn't quite come down in cost compared to transcriptomics and that sort of thing.
Thanks.
I have another question. Am I allowed, Kenny?
Marion did have her hand up, but you're going, so.
Well, just a quick one, Jenny. What really struck me about the step change versus the 1.5 degrees versus the 3 degrees, and looking about, first of all, global warming and ocean warming with respect to the gradual warming, and then you have the insults where you have heat waves. You've almost got two different warming scenarios. And so in just putting and now we're not in El Nino anymore. Going forward, how do you think these fish would cope, for instance, with a step change of 1.5 degrees versus a heat wave thing, which would be, they'd be slammed by 3 degrees?
So that is really exciting because that experiment is a new PhD student as a part of my future fellowship. So I'll have the answers in a few years. But I think the sort of heat wave exposure would bring in a whole different suite of mechanisms and different physiological responses that would be really interesting to understand, because I do think that shifting to deal with a continual warming may not suit you, may not be suitable then when you have to experience a heat wave on top of that. And that might be the real nail in the coffin in terms of, yeah, it's fine if everything just gradually warms and doesn't fluctuate too much. But I alluded to it a little bit. I didn't want to give it all away because this is really Rachel's and Lucrezia's PhD. But the interesting thing when they did it again, we've done the experiment again, but better, hindsight, start over, add in the diurnal cycles. Diurnal cycles on plus 1.5 is a lot tougher, and it does make a difference. So it does make a difference how we simulate and making an effort to really add in these natural variations and like you said, heat waves on top of normal warming or on top of expected warming, I guess, and continual exposure to heat waves, whether, like I say, whether they'll be better primed for the next one, or did it come at a cost that then means the next time is a real problem. So yeah, there's a lot to do in that space for me, I think.
Yeah, thanks, Jenny.
And I think Heather's typed into the chat to back me up. Thanks, Heather.
Marion, did you still have a question there?
Yeah, hi Jenny, thanks for the talk. My question is genetics. I was just wondering, because these fish are biparental carers, right? So when you actually put these offspring under these different conditions and whatever, I assume you're rearing them in the absence of their adults. Or...
No. So basically we leave them with the parents. So one of the negatives is that the embryonic development ends up being linked to the parental development, and we can't separate the two. So basically we decided that we would rather have good parental care, good survival, sort of natural situations there. And that's why we've made the effort at times to try and switch and have this different reproductive environment. So pairs that were developed in warming, but then reproduced back at control conditions. And we have done a little bit trying to incubate the eggs ourselves, but that becomes quite labor intensive and very difficult to manage. So we've used that with a minor project student last year, more to try and understand whether experiencing warming during different developmental stages of embryogenesis has an effect rather than anything else.
Yeah, no, I actually think it's good to rear them in a more naturalistic setting with the breeding pair, but whether or not you could actually do things like quantify parental care behaviors, or if you've done that already, potentially correlating that with their offspring's, various physiological traits, and maybe also looking at hormone profiles to try and better understand the mechanisms of the reproductive, whole reproductive side of things.
Yeah, so definitely when we've tried to investigate the hormone side of things, there's a lot of variation, and acanthochromis is good, but often it's very hard to try and get enough clutches to work out their cycle, so then you can catch the adults right at the right time of the reproductive cycle, so you don't have so much variation in testosterone and estradiol and cortisol. But in terms of this parent side of things, that's definitely something that Yogi, that was on one of the later slides, was really interested in for his honors, and now he's coming back for a PhD. I think that's something we really do want to look at, especially because if water temperature is warmer, you might expect parents to spend more time fanning to try and get better oxygenation over the clutch, and some really interesting things there that could just be some of the effects you see are that certain parents are maybe not doing as much to maintain the clutch in those warmer temperatures. So then it ends up being more of a parental effect than a truly transgenerational one, but still really interesting. So I think the reproductive side of things is definitely something that needs more time, because it seems like it'll be so important.
Yeah, definitely. And with parental care, also the amount of time the pair has spent together already often plays a major role. I know there's been some work with Amphiprion showing that essentially that greater familiarity between the breeding pair influences essentially their reproductive output and success. So yeah, that'll be really cool to look at.
Yeah, and definitely watch this space for when Rachel's research comes out on the maternal and paternal contributions, because there's some really interesting stuff when mothers and fathers haven't had the same environmental conditions. So it does seem like the partnership could be pretty important in terms of what you then get in terms of the offspring generation.
You're doing really well with the question time here. I think Sarah's up next.
Thanks for that presentation, Jenny. That was really fascinating, and I'm slightly concerned that my question is going to be a bit ignorant, because some of your presentation definitely went over my head. But it did get me wondering about the timescales over which plasticity shows itself through reef fish, and how that lines up in terms of the practical implications for getting people to study them. For instance, some of your slides kind of left me with the impression that we've got about 3 generations in a couple of years, and that strikes me as a limited opportunity within the course of a PhD candidature, for example. That are there, I suppose 2 questions. How long does it take for plasticity to show itself and be detectable? And also that got me wondering, if you're committing to a particular family line, how long do those commitments last for? What's the longest length of time that somebody's followed a family line and seen things like plasticity roll out?
So I think you've touched on something important, that coral reef fish in general as like a model for plasticity are challenging because of the generation time. So there's been work on, say, C. elegans and Drosophila and others that have really high turnover that can answer questions about like the length of time that things will continue. So an experience in one generation and how long it'll last after. So they can get up to sort of, decent generation times 7, 10 more. But for us, we've never gone past 3, the 3rd generation. So we have the 3rd generation in the lab again. And after that, it gets really hard because it was always difficult to start with a big enough genetic diversity in the first place. So in the experiments we've just started again, Rachel's work was really focused on not starting with a heap of families, but so that we could really tease out and do very structured pairings to understand other aspects in more detail. But then by the time you get to the third generation, you've not got enough diversity to keep pairing everybody up without possibly ending up a little bit incestuous in terms of your understanding. So I think for me, there's other model fish that we would have to go into, probably over coral reef fish, like the freshwater species are just more amenable with the much shorter generation time. So, you know, you go down to sort of maturity within 40 days rather than, you know, it's every 2 years for a generation in this one. And if we switch to something like perculate, it'd probably be a minimum of 3 years. And, you know, it gets longer and longer with other species. And what was your first part of the question, sorry?
No, I think that covered it. That's okay. I was just wondering how long it takes plasticity to be apparent, whether it's 1, 2, 3 generations, and how that sits in relation to carrying out research.
Yeah, I think, I think you can get within generational plasticity quite quickly. So most of the developmental plasticity, other species that we've talked about, we sort of just did a couple months in early life. I guess you always have to kind of do like a short period or a whole year, because then you end up back at summer, or a whole nother year, because then you end up back at summer. So we're always kind of trying to test at summer where we think it's going to be the most important. So if you don't do it in the early life, then you have to wait till 1 year of age, I guess. And I think the manifestation of plasticity is really interesting in terms of theory versus practice. And there's a lot of theories out there on environmental variation, you know, so actually, if the environment varies greatly within a generation, you'd expect reversible. If it varies a lot between generations and it's unpredictable, you expect developmental. And if it varies a lot between generations and it is predictable, then you expect transgenerational to evolve. But often we don't have very good tests for all these sort of expectations. Yeah, I think there's a lot more work to do, and it's great to do them in non-model organisms as well, because it does, I guess, test the generality of whether what we're doing in some of these clonal species in the lab for plasticity and evolution research could ever be applied to the complexity and diversity that we do have in some of these other systems. So yeah, it does take a while. And that can be challenging on like funding cycles as well. Like, so, you know, you often kind of like plan a project and then, you know, shut it down. I guess I've always been really fortunate that to have support from people like Phil and my supervisor, my previous PhD supervisor, Mark, who would help me keep things going past, say, a funding cycle or a PhD cycle, and so that I didn't have to just make a hard decision. So I've been really lucky with that.
Thanks, Jenny.
Well, we're coming close to time now, so I might wrap that up unless anyone has one last quick question to race through with. But I think there's been a few big onslaughts of questions here for Jenny, and I'd like to thank you again for coming as our ACRS Fellow for 2020, and hopefully we can all see each other soon.
Thanks, everybody. Merry Christmas.